DNA, or deoxyribonucleic acid, is in every living organism and contains our genetic code. From our height to our eye color, DNA is what is responsible for making us who we are. As you can see by this drawing, DNA is composed of rungs and side rails. The rungs are made of nitrogenous bases: adenine, thymine, cytosine, and guanine. The side rails are composed of phosphate backbones, a sugar molecule, and a phosphate group. As mentioned before, the sugar that constitutes DNA is deoxyribose. In RNA, the sugar is called ribose. Looking at these two molecules, the difference is the lack of an oxygen atom at the second carbon. This is why DNA is called deoxyribose. It is important to make the distinction because while RNA and DNA are both nucleic acids, they serve different functions in the cell. DNA serves as the genetic information of the organism, while RNA helps express this information by assisting in protein synthesis. The phosphate, sugar, and base will come together into what is called a nucleotide. Base pairing is very specific. Adenine will always pair with thymine, and cytosine will always pair with guanine. Cytosine and guanine have 3 hydrogen bonds, while adenine and thymine have 2. The hydrogen bonding is important for DNA strand stability. However, base stacking is the main force that helps stabilize the DNA's double helix. This is due to the van der Waals and dipole-dipole forces between the hydrophobic bases. Base stacking helps prevent the hydrophobic bases from interacting with water molecules by stabilizing the overall 3D structure. DNA helices have major and minor grooves. The major groove is where the phosphate backbones are furthest apart. The minor grooves are where the phosphate backbones are close together. Lastly, DNA strands can be left or right-handed. In the Watson and Crick DNA model, the most common model, DNA is right-handed with approximately 10 base pairs per turn. This means that there are about 5 rungs found between each twist in the helix. Left-handed DNA strands are much more rare than right-handed DNA strands. So how do we determine if we are looking at a left versus right-handed DNA strand? Here are 2 birthday candles to help explain the idea. See how the lines point toward the right? This means this one is right-handed, versus this one. These lines point towards the left. That means it would be considered a left-handed DNA strand. As stated before, left-handed DNA strands are very rare. This doesn't stop the public from using left-handed DNA images all the time. Here are some examples of left-handed DNA strands found in recent internet articles and advertisements. As you can see, all of the phosphate backbones point toward the left. I hope these couple of images help you see the difference between right and left-handed DNA strands. I also hope you learned something about the DNA structure, from its composition of its nucleotides to maybe something new about its shape.